this multi-scale topic with a talk that will be more conceptual and with very little of data. The data, I hope, will come maybe in the next meeting because we are still accumulating them. I'm going to talk about strategies that we can use today, and they are improving to handle what we call the root hydraulic architecture, and I will explain you what we mean by that, and to address that at multiple scales. So, as you understood, the story that I'm going to cover is about the efficient capture of water, which is one important aspect in phenotyping, especially for drought tolerance. And it's also very challenging to have an idea of what is actually occurring underground. So this topic has been largely studied until now with what I would call an architectural view. And when we speak about the steep, deep, and cheap ideotype, something like root depth and root angle, that has been well validated in a number of soil and climate scenarios, you understand what I mean by an architectural view. Now, in the framework of the evolution of the progress in the technologies and our understanding of how the biological system work, I would like to address whether phenomics, let's call it like that, brings us new opportunities. So we have a number of technical improvement, and we have seen yesterday in the root workshop the novel platforms that are now available. I will integrate into phenomics, maybe I shouldn't, but data on gene expression, and typically something that is going on in Louvain is measuring the aquaporine expression in maize genotypes, and there is an heritability for this trait and genetic variability. And we also have, but that's long in the past, anatomical data like xylem vessels, and all of these are, as you understand, really involved in the capture of water by plants. So I will start from the field, and maybe Graham will be happy, but I depend if I explain this well. But today, root water uptake in crop model, and this reflects quite well our level of understanding, is a matter of over a number of soil layers. You will have the soil water availability times an occupancy factor that goes from 0 to 1, that tells how much of the soil volume is actually occupied by roots. So this is typically related to architecture. And then you also have another term, which is here KL, which is a mixture of soil effects, the soil conductivity, how easy the soil is ready to keep the water to the plants, and at the same time, how aggressive the plant can be to actually get this water into the roots. So the KL is a mixture of root hydraulic conductivity and a mixture of soil hydraulic conductivity. And so this approach allows to integrate based on the architectural view, differences of root angle between two different genotypes, a wide angle genotype and a narrow root angle genotypes, that is mainly reflected through the occupancy factor. So now I will come to illustrate a little bit the limits of this architectural view, and starting by this illustration, you have on the ordinate the hydraulic conductivity, and on the axis the water potential in the soil, and we represent here in green is the range of variability for the hydraulic conductivity of the root material, and in purple you have the hydraulic conductivity of the soil, and this one is actually affected by the soil water potential. So in the context of a drying soil, so a drought episode that develops, you start from a situation where the soil is more conductive than the root, which means that the root is actually deciding where the water is actually taken up. And this is where architecture is deciding for you. While as the soil dries out, progressively the root becomes more conductive than the soil, which means that the soil is telling where the water is actually available. So there is a change in strategy from the plant point of view that you need to address if you are actually willing to exploit this in a very efficient way. So now I come to a second idea of the limits to the architectural view, which is more that there is more opportunities for breeding than only the architecture. And if you move from the left to the right of the slide, we basically follow the path of water from the bulk soil through the rhizosphere, entering the roots, moving long distance through the xylem and ending up in the shoot. I'm sorry if I stop my talk to the shoot, but someone else is taking over, of course. What happens in the bulk soil is long distance transport, gradual, slow, with temporarily gradual transition. You don't have a sudden change of water potential over time in the soil. And bulk soil, this part of the process is mainly driven by root architecture. Where is the bulk soil is actually relative to where you place your roots. Then the water enters the rhizosphere. In the rhizosphere, you will have transient, really rapid changes. You can have loss of conductivity that occurs in the rhizosphere, and you have also very sharp transition of water potential and speed. So this is a place where actually the roots can also change the conditions by producing exudates or whatever kind of material that you can find in the rhizosphere. Then you move to the roots. Crossing the roots is obviously something where the roots has something to say. Well, we will say now aquaporins, probably apoplastic barriers have a definite role in determining the hydraulic conductivity there. Then the xylem, well, xylem, it's easy, it's a pipe, so the diameter of the pipe should be enough, but you have a number of transient phenomena like cavitation that actually make your life a little bit complicated. So the point is, the architectural view only focuses on the access to the bulk soil, while there is a possibility to alter what happens in the rhizosphere, the aquaporine expression, development of apoplastic barriers are all opportunities to actually also modify the uptake of water. So to study that, I will illustrate here with the use of a model that is called RSWIM that has been developed in Jülich and in Louvain by Mathieu Javot and colleagues, but it's kind of similar to OpenSynRoop is doing the same sort of things, which basically represents with the electrical analogy the soil system, the plant system, and the connection between all of them. So the output that you get with this sort of very detailed models that can take hours to run because there are a lot of equations to solve. You have on the first side, you have the root system architecture that you can get from models or from a phenotyping platform. Then, wow, time is running fast. Then we have the water content distribution. A very important term you can extract from this is the sink terms, is where where water is actually taken up by your root system, and then you can also extract the xylem water potential. It is probably very important to capture a physiological aspect of this, and also the path of water that will be important if you want to simulate long-term water movement in your soil. So the good news is this model is really complex, takes hours to run, but Valentin Couvreur has been able to identify, by starting from the equation of the model, to simplify them and found an easy analytical solution that is shown here, and which basically is equivalent to the equation that you have in APSIM. But no, it's not a KL times occupancy. It's a different way where you really discriminate and distinguish the two biological components. So you have on the one side the SSF, or the standard water uptake, which is based only on the root architecture, and which would be how much of water is taken up if the soil water potential was constant. And then on the other side, you have this compensatory water uptake, which is how much of deviation compared to the standard water uptake is actually done due to, on one side, the root architecture, and on the other side, the redistribution of hydraulic conductivities within the root system. So, I try to do the rest in 2 minutes. So we had also used this model for a very interesting prediction that relates more to aquaporins and xylem vessels. Is trying to simulate and to predict the uptake of water as a function of depth. Okay, in 3 different scenarios, left, middle, and right. Left is a low ratio of radial conductivity of the root to the axial conductivity. So you could simplify and say radial conductivity is large if you have large aquaporin expression. That's an idea. We can test this, but we can. While on the right side, you have a large Kr over Kx, which means a high expression of aquaporins and probably relatively low conductivity in the xylem. What you see is that on the left-hand side, your distribution of uptake is kind of homogeneous and really well distributed over the whole depth of the profile. While on the right-hand side, if you increase the radial conductivity of the root system, water is largely taken up on top of the soil, and then the water is not at all taken in the deep profile. So this is probably a way, without touching at architecture, to generate a water saving strategy where you decide to take up water at different places. And it does not involve root architecture at all. So we wanted to try to understand this a little bit, and at least to play a little bit with this concept. So we went back to the literature looking at what regulates this root hydraulic conductivity. And so you know the classical story with the different pathways. And we have decided to create a model where we capture the structure of the root tissues, the position of the apoplastic barriers, and the level of aquaporin expression. So this leads to this type of model where 4 different cells, you see in gray the cell wall, in light blue the vacuole and the inside of the cell, and all the different pathways through plasmodesmata, through the cell wall, through the membranes are actually simulated. So this is just with 4 cells. So if we apply this to real cross-section of, this is a cross-section from maize that we segmented using the CellSet software, we were able to simulate the membrane flow density, the plasmodesmata flow density, the wall flow density. So we have the flow of water into all of these compartments, and this is just a zoom up. If you want to know more, you can come to me and I can explain you, but it's just the fancy picture at the moment. From this, we are able to simulate how much of the water flow from the outer layers to the inner layers goes through the apoplast, through the symplast, through the plasmodesmata, and you see 2 different contrasting scenarios if you have or if you do not have an exodermis, for example. And so these allow us to estimate the segment level radial conductivity, which was the higher properties. So, as a summary, we have a biological situation where that's based on our understanding of biologies. We have a regulation at the I try to catch the mouse. We have a regulation at the cell level. Then you aggregate this by adding different cells to have the pathway of water across the roots, and then you can stack those different root segments into a whole root, and then you can stack the roots into a complete root system architecture. Now what we have done is we have done this mathematical aggregation by going from the cell to the radial section and from the root going to the root system using these 2 models that I showed you. And what we have also done is find analytical solution, that means straightforward, that describe the imaging properties in a set of scenario. So this is a good news because it means that now we can more easily try to think about getting aquaporin data in a crop model without actually changing much inside the crop model and without talking about a complete functional structural plant model. So that's it. Maybe just the first sentence I will keep as a conclusion. That's probably might be the question that Graham will not ask. Is the architectural view works and do we really need more? So we see that there is a potential. We can start to make link, identifying at the right scale, the right properties, and then scale them up. So this is possible. At this stage, it remains a model, and we start to have to gather a lot of data that will allow us potentially to the truth. Thank you very much.
Thank you, Javier. I think that...
I apologize for the...
No, that's okay. I think we have time for only half of a minute question. If you have any burning question to Xavier.
I will be at the coffee break. Please.
So I think you showed initially that when the radial conductivity was high, in that architecture profile, in that soil profile, the water uptake was higher from the surface layers compared to when the axial conductivity was high. So that right there. Yeah. So so now to go to the aquaporin and the anatomical reason for this. Is this because the root lengths are the same, and the root lengths in these surface areas somehow have higher expression of the aquaporins and less radial barriers to the water movement? And can you explain that?
Okay. At this stage, with what we have in the model is an analogy with a pipe that would be slightly porous or not. And if you take a porous pipe and you apply a suction at a certain level, and if you have the same pipe which is less porous, if the pipe is less porous, water will enter through all positions along the pipe. While if the pipe is largely porous, water will follow the closest path, and so you will take water most from the top. There is no aquaporin notion or whatever. This is pure hydraulic here. If you take a pipe with holes in it, depending on the density and the number of holes, you will have this sort of distribution. So this is a pure hydraulic phenomenon. So now, what we try to do is to relate the data like aquaporin expression to the radial conductivities. So if you have the ability to measure the water uptake, you will be able to tell whether you are on the left or on the right. It will tell you what is the Kr over Kx ratio. And then you can go back to your plant material to see whether this might be related to aquaporin, but so it's the other way around. This model is only an hydraulic model. It would be exactly the same that would be used in building house and make sure that you have the right pressure going when you open the tap in the morning. Okay?